So now let's look at the formation of some isomers, two isomers of glucose, and discuss how that will be important in the formation of different starches. And I want to re-emphasize that right now we're kind of going through nomenclature. We're getting under our belts the important knowledge of what the major macromolecules in biological systems are. In some ways, I don't think it's that boring, but it's not as exciting as when we can get to some of the interesting processes involved in energy flow and the decoding of genetic information and gene regulation and the experiments that allowed us to decipher those complex processes. But right now we're just kind of learning the lay of the land in terms of the molecules that are found in cells, and it will all become important. So this is, although rather dry, it's important that we understand all this. So here we have glucose, and in solution, glucose will actually form a ring. It's a linear molecule, but it will form a ring, and what essentially will happen is that this oxygen will attack that carbon, and by attack I mean it will form a covalent bond with that carbon. That is energetically favorable. Oxygens often do that. They attack other atoms and form bonds with them. And so that oxygen is bonding to that carbon, that number one carbon, this oxygen is. And remembering this oxygen that is doing the attack on the number one carbon is also bonded to the fifth carbon. And we know that oxygen is only capable of forming two bonds. So in order for this oxygen to bond with this carbon, it donates its hydrogen to the oxygen that was doubly bonded to that carbon. And that oxygen then can only form a single bond to that carbon, and now forms a single bond to the hydrogen donated by the attacking oxygen. So the carbonyl oxygen that is attached to the first carbon, when this cyclization occurs, when this ring formation occurs, that carbonyl oxygen acquires the hydrogen from the attacking oxygen. Now, depending upon how the resulting hydroxyl group is arranged with respect to the first carbon, whether it is above or below the plane of the ring, above here and below there, that defines two isomers of glucose, alpha glucose or beta glucose. And those two different forms of glucose are used differentially by different enzymes that polymerize glucose into starches and long-chain carbohydrates. And we've already talked about how glucose is a structural isomer of the fruit sugar fructose and is a stereoisomer of the sugar galactose. All of these are monosaccharides, that is single sugars. Now disaccharides are two sugars linked together. So for example, we can combine in a condensation reaction or a dehydration reaction alpha glucose and fructose together to generate the sugar sucrose. You've all heard of sucrose, I'm sure. Or maltose can be generated by the linking together of two glucose molecules together. Two glucoses will equal a maltose when joined. And these are just examples. Now, when monosaccharides are polymerized into disaccharides and even longer chain polysaccharides, when that happens, those polymers are used to store energy, whereas single monosaccharides are usually the entry point into metabolism to be broken down to derive energy from them. So glucose, for example, is the sugar that enters the pathway called glycolysis that we'll talk about later. That is the first step in breaking down sugars for energy. But when polymerization of monosaccharides occurs, the disaccharides and resulting longer chain polysaccharides are used to store energy. So now let's look at some of these polysaccharides, these longer chain saccharides, a higher number than just higher than 2. So polysaccharides. They are long chains of monosaccharides that are linked, like disaccharides are, through dehydration or condensation synthesis. And they are used for energy storage. Plants use starch and animals use glycogen. They're very similar. And plants also use long-chain polysaccharides for structural support in the form of cellulose. So as we will see, beta-glucose is used to form long fibers, to be polymerized into long fibers of cellulose, which basically are why trees can stand as tall as they do. And arthropods and fungi both. So we have the invertebrate arthropods, and fungi use chitin as their structural support. And chitin is a polymer of modified glucose. That glucose is called N-glucosamine, and that's polymerized into chitin. So let's look at how glucose becomes polymerized into polysaccharides in plants and animals. So here is alpha glucose, and there you can link alpha glucose molecules together by linking the 1 carbon of one glucose molecule to the 4 carbon of another molecule, as shown here. Here we have 1, 4 linkages, 1, 4 linkage, 1, 4 linkage. And the oxygen on the hydroxyl group on the 1 carbon ends up forming a bridge between the monomers in the polymer. Also, there can be a 1,6 linkage. So you can create branched chains, not just long single chains, but you can create branched chains by linking glucose molecules, glucose monomers that are part of a long polymer. Another glucose can come in and be linked to the carbon here, the 1, 2, 3, 4, 5, 6th carbon in the chain, in the ring rather. So here we have a 1,6 linkage, 1,6 linkage, and that can form branched long chain polysaccharides. So here in plants we have 2 storage forms of glucose, of alpha glucose, amylose and amylopectin. Amylopectin consists of branched chain polysaccharides, whereas amylose is a single long chain. And there are different percentages of these 2 different starches in plants, different parts of plants. So amylose and amylopectin are plant polysaccharides used for energy storage, and then these can be broken down into monosaccharides by hydrolysis, and that hydrolysis produces monosaccharides that can then be broken down further to yield energy, as we will study later. And in animals we have glycogen, which are much longer branched chained alpha glucose linkages than in amylose and amylopectin. And here are these glycogen storage depots in an animal cell. And here is a scanning electron micrograph showing aggregates of amylose and amylopectin in plants. Now, those were alpha glucose polysaccharides. What about beta glucose polysaccharides? They do not form branch chains. And in plants, 1,4 linkages of beta glucose yield long chains of single chains of a polymer of linked glucose molecules. And that is what comprises cellulose. And unlike the starches and the glycogens that we talked about, which are alpha glucose polymers, polymers of alpha glucose, those can be broken down, as I've pointed out, quite easily to yield monosaccharides that can be used for metabolism to derive energy from them. Unlike the alpha glucose starches, the cellulose, which consists of beta glucose, is very difficult to break down. And in fact, that's why digestion of roughage, of plant roughage, can be very difficult. And for example, cows need bacteria, symbiotic bacteria that live in their gut rumen, that those bacteria produce enzymes that are capable of breaking down cellulose. The ungulate, the cow, would not be able to do that on its own. It needs those bacteria. So cellulose is a very tough, fibrous material, provides structural support for plants. Well, now we've covered carbohydrates thoroughly. Let's move on to nucleic acids, the informational molecules of the cell. So nucleic acids are macromolecules. They're polymers of nucleotides, and those nucleic acids are built from monomeric nucleotides. And a nucleotide contains a sugar plus a phosphate plus a nitrogenous base. Excuse me. I'll show you an example in a moment. Sugar is called deoxyribose in DNA. The sugar in the nucleotide is deoxyribose in DNA, or ribose in RNA. And the ribose sugar in RNA differs from the deoxyribose sugar that is present in the nucleotides of DNA because it contains a hydroxyl group on its number 2 carbon, whereas in the nucleotides in DNA do not contain such a hydroxyl group on the 2nd carbon of the ribose sugar in the nucleotides of DNA. So the nitrogenous bases that are parts of the nucleotide, we have 3 components here: sugar, phosphate, nitrogenous base. Nitrogenous bases are of 2 types. They are purines and pyrimidines. The purines are adenine and guanine, A and G we can refer to them to. And the pyrimidines are thymine, cytosine, and uracil. Uracil is found in RNA, and thymine is found in DNA. But otherwise, adenine, guanine, and cytosine are found in both DNA and RNA. We'll talk about that more as time goes along. Now, the nucleotides are connected by phosphodiester bonds, and I'm going to show you what that is in just a moment. But here is a nucleotide that is could be found in both DNA and RNA. The difference in the ribose sugar, here we have a ribose sugar here. It has 5 carbons, 1, 2, 3, 4, 5, this 5th carbon here, and an oxygen at the apex of this sugar, if you will. So it's a ring sugar. And in DNA, the 2nd carbon, we call it the 2' carbon, does not have a hydroxyl group. It just has a hydrogen on it. But in RNA, there is a hydroxyl group attached to this 2' carbon. And the other difference is that in terms of the possible nitrogenous bases that are in nucleotides in DNA, there's A, T, G, and C, adenine, thymine, guanine, and cytosine, whereas in RNA, uracil replaces thymine. And then attached to the 5' carbon of the ribose sugar is a phosphate group. So we have 3 components to the monomers that will build up either RNA or DNA. Nitrogenous base part, and that is linked through what we call a glycosidic bond to the 1st carbon in the ribose sugar. We have a ribose sugar then here, and then a phosphate linked to that 5' carbon. And this is called an ester linkage because it's through an oxygen. So this carbon is linked to an oxygen, is bonded to an oxygen, which is then bonded to the phosphorus atom of the phosphate group. And that is, those oxygen bonds are ester bonds. So in a moment, we'll see why the DNA and RNA molecules are said to be held together by phosphodiester bonds. So I'm going to now talk about how the monomeric nucleotides are linked together in a chain for both DNA and RNA. We mentioned that we're going to specify phosphodiester bonds as the linkage between nucleotides. And we'll talk about that in the beginning part of the next section. But right now I'm just going to leave you with a little view of kind of the beauty of DNA. It's a really beautiful molecule, the double helix, 2 intertwined strands of DNA. That is the genetic material. So I'll leave you with that, and we'll pick up in the next part with the bonding arrangement that is present in both DNA and RNA.